Good morning, ladies and gentlemen. I am Samia Zahoor. I warmly welcome you all on the 8th International Conference on Applied Zoology 2025 on the behalf of Organizing Committee ICAZ. I will be the moderator of this session, which include talks from the field of fisheries, biochemistry, and toxicology, highlighting the diverse and significant research contributions from scholars across various institutions. So let's begin our session with the talk of Ms. Labadla Shaima from Mila University Center, Algeria. She will be presenting her research on structure and seasonal dynamics of avian communities in Beni Haroun Dam, an artificial wetland in northeastern Algeria. Please welcome Ms. Labadla Shaima.
Good morning, distinguished organizer, respected professors, and fellow researchers. I am Labad Dashema, a PhD student at the University Center of Mila, Algeria. It is a great honor for me to take part in the 18th International Conference on Applied Zoology 2025 and to present my research today. So the title of my presentation is Structure and Seasonal Dynamics of Avian Communities in Beni Haroun Dam, an artificial wetland in northeastern Algeria. So the wetlands, natural and artificial, are vital ecosystems that support diverse species, notably migratory waterbirds. This area provides essential nesting, feeding, and resting sites for birds during their long migration between breeding and wintering grounds. While Algeria's natural wetlands have been well studied for their role in supporting waterbird ecology, artificial wetlands, which may serve as a key refuge during drought, remain under-researched. To better understand the significance of artificial wetlands for species like the great cormorants, it's important to define the waterbird and their relationship with wetland ecosystems. So the waterbirds are migratory species uniquely adapted to wetlands ecosystems, which provide them with essential resources such as a food, a shelter, and breeding sites. This bird depend on both natural and artificial wetlands, particularly during migration and wintering seasons. The role of this habitat is therefore crucial as they offer a vital stopover points that ensure a survival during long-distance migrations. Studying these environments is essential for understanding and conserving waterbird populations, highlighting the importance of wetland preservation and sustainable management. The wilaya of Mila is located in the eastern part of Algeria. So this wilaya is at the northeastern part of Algeria at an altitude of 464 meters and 70 kilometers from the Mediterranean Sea. The Beni Haroun Dam is the large and strategic hydraulic complex in Algeria. Now moving to the methodology of this study, which outlines how the data was collected and analyzed to assess the ecology of the great cormorant and artificial wetland of Mila. For this research, 3 observation stations were strategically established along the dam's shores, choosing for their accessibility, broad vantage points, and comprehensive view of bird population across the site. Its station included multiple observation points to ensure an adequate survey of avifauna. Bird counts were conducted twice a month at all study sites using direct observation methods aided by a binocular, a telescope, a camera, and the Hensel guide for accurate species identification and monitoring throughout the dam area. Now moving to the results and the discussion. The family of Phalacrocuracidae, which include the cormorant, is undoubtedly the most remarkable, and the Bniharon dam represented a significant value of 34.87%. The Anatidae family at the dam is primarily respected by 9 wintering species, with 8.51% of the population. The Radidae family is dominated by 26.27% as the most abundant and resident species. Other bird families show minimal presence. This picture represents a P-share illustrating the proportions of wintering bird families at the Bniharon Dam observed from October 2023 to March 2024. And this picture represents the evolution of the population of the Phalacrocuracidae family wintering at the Bniharon Dam from October 2023 to March 2024. Monthly population counts of cormorants show a clear seasonal trend peaking in January with over 1,200 individuals. Numbers rise from October, reach a maximum in early January, then decline significantly in February and March. This pattern suggests a seasonal wintering behavior, with the cormorants arriving in the colder months and disappearing as temperature rises, likely due to migration. Having discussed the methodology and findings, we now arrive at the conclusion of this study. The Bniharon Dam is crucial for avian biodiversity, especially for migratory species that rely on this site during the winter. This study highlights the need to protect and sustainably manage artificial ecosystems like this, as they provide essential habitat and support regional biodiversity. I would like to thank you all for your attention, and I hope this study has shed light on the ecological value of artificial wetlands. I am now happy to answer any question you may have. Thank you.
Thank you, Miss, for that insightful presentation on avian community dynamics. Next, we have Dr. Shagufta Saeed from the University of Veterinary and Animal Sciences presenting her work on evaluation of synergistic anti-diabetic potential of Prunus persica, leaf extract, and chromium in alloxan-induced diabetic rats. Let's welcome Dr. Shagufta Saeed.
Assalamualaikum. I am Dr. Shweta Said from the Institute of Biochemistry and Biotechnology, University of Veterinary and Animal Sciences, Lahore. First of all, thank you very much for inviting me in the 8th International Conference on Applied Zoology. The topic of my presentation is evaluation of synergistic anti-diabetic potential of Prunus persica leaf extract and chromium in alloxan-induced diabetic rats. First of all, introduction. Diabetes mellitus is a metabolic syndrome characterized by elevated blood glucose level caused by insulin-impaired secretion or action. Pakistan ranked first globally in diabetic prevalence. Although the treatment of diabetes has improved over the past decades, its mortality and mortality rate remains high. Prunus persica, it belongs to family Rosaceae, and its genus is Prunus. Common name is peach or arb, and its habitat is a native of Asia, specifically northwest China. In Pakistan, it is found in KPK, Swat, Balochistan, and the northern areas. Next is the chromium. It is beneficial trace element for the human body and it increases the insulin sensitivity and regulate blood glucose levels. Chromium picolinate widely used as a nutritional supplement. Statement of problem and diabetes rate is increasing in worldwide while many drugs that are currently being used come with significant side effects. The use of presently available drug for its treatment can lead to toxicity or gastrointestinal problem if used for a longer period. To overcome the adverse side effect of these drugs, there is a need to develop natural remedies that have anti-diabetic potential and are safer to use. Hypothesis, Pannus persica leaf extract along with chromium has anti-diabetic effect in alloxan-induced rats. Objective is to investigate the anti-diabetic potential of Pannus persica leaf extract and chromium supplements separately and synergistically to study the histopathology of pancreas in diabetic rats after treatment with Pannus persica leaf extract and chromium. This is a methodology. First step collection and identification of plant, and second one is extract preparation, and then phytochemical analysis of the extract was performed, and then experimental animal grouping and diabetic induction, then extract administration and biochemical and histopathological analysis, and at the end statistical analysis was performed. Experimental station experiment was carried out at Institute of Biochemistry at University of Veterinary and Animal Sciences, Lahore, and plant leaves was collected from the Abbas Nursery Farm, Lahore, and Tanna Farm House, Sabah. Plant identification was carried out from the Department of Botany from GC University, Lahore. Extract preparation was done in the following step. First of all, 150 gram of dried powder dissolved in 900 ml of distilled water and then kept the mixture at 37 degrees centigrade in shaking incubator for 3 days. Collected the filtrate using Workman filter paper number 1 and extract evaporated to dryness in the water bath. This is the steps how extract was prepared. And then phytochemical analysis was performed. GCMS analysis was performed for identification of phytochemicals in extract by TT Labs, Lahore. Experimental animals, visitor mirrors of 150 to 200 gram body weight were selected, and the fresh water and standard red diet was provided to rat at Lipton. Diabetes induction and rats were kept on overnight fasting condition with free access to water and then peritoneally injected with alloxan's monohydrate and glucose level was checked after 72 hours of injection and rats were fasting glucose level above 180 milligram per deciliter were selected for further study. These are the grouping of experimental animals, total 40 rats that are divided into each 5 groups and into 8 groups and each group contained 5 rats. These are the rat house in plastic cage and wooden litter and grouping of experimental rats. These are the parameters. Number 1 is body weight, and second one is fasting blood glucose level and biochemical analysis. And in the biochemical analysis, serum insulin level and serum amylase level will be checked. This one is a blood sugar estimation using glucometer. And in the Picture B, blood sample was collected by a cardiac puncture method and in the C, blood sample in the clot activator valve. These are the histopathological analysis. The pancreas were removed, 1 rat per group and fixed in 10% formalin solution for the histopathological study. Section of 5 to 6 micrometer thickness was cut and stained by hematoxylin and eosin for visualization under the light microscope. Then statistical analysis Significant difference between experimental group were statistical analysis by one-way ANOVA using SPSS software. Results, and these are the results of body weight. As we can see that the body weight in day 1st, day 7, day 14, and day 21 was recorded, and the group 6 has noted that increase in body weight after 1, 7, 14, and 21 days. These are the histograms showing the comparison of body weight in terms of mean standard deviation among the all groups. Then hypoglycemic activity, blood glucose level. The comparison of blood glucose level in control and treatment group was performed. And at group 6, we can see that after the 1, 7, 14, and 21 days, the glucose level is gradually decreasing. This is the histogram showing the change in blood glucose level over the first 7, 14, and 21 days. These are the results of biochemical parameters, serum amylase level in the control and treatment group. And in the group 6, we can see that there is a high level of serum amylase. This is the histogram showing the comparison of serum amylase level in terms of mean standard deviation. Then another biochemical parameter, serum insulin level in the control and treatment group. And here we can also see that in the group 6, the serum insulin level is recorded high. This is the histogram showing the comparison of serum insulin level in terms of mean and standard deviation. This is the histopathological analysis, and figure A, that is showing the group 1 negative control and group 2 that is positive control. And in the C, that is group 3 diabetic control slide, and group D, that is group 4, that is given 250 milligram per kilogram of dose, and each slide indicates the 500 milligram per kilogram, and group 6, that is 100 milligram per kilogram, and then in G, group 7, that is 1000 milligram per kg, and in the group 8, that is the histopathological analysis of the group 8. These are the GCMS analysis result of aqueous extract of Parnas persica, and different waves, peaks indicate the different phytochemical present in the leaf extract. These are the bioactive compound found in the aqueous extract of Parnas persica leaves. Mandelic acid 3-hydroxy 1-pyrazole 4, acetic acid, lolylide, cis, snipyl alcohol, myristic acid, carbachol, squalene, and then conclusion. The present studies show that Parnas persica leaf extract in combination with chromium picolinate offers significant protective effect against alloxan-induced pancreatic beta cell toxicity in a dose-dependent manner, with highest dose 1000 milligram per kg in chromium and 20 microgram per kg show the most effective result, and this combination proved to be the more effective than either treatment alone. Highlighting a clear synergetic effect. Furthermore, the study identified and reported the presence of various phytochemicals in Parnas persica leaves, which was not studied before, suggesting their possible role in modulating blood glucose level through multiple mechanisms. This is all about my presentation. Thank you.
Thank you, Dr. Shagufta, for sharing your valuable biochemical insights. Moving towards our next presenter is Ms. Sadia Habib from the University of Agriculture, Faisalabad. Her topic is microbial analysis and hematological parameters of labio rosita after dietary administration of black cardamom of different percentages. Please welcome Ms. Sadia Habib.
Assalamu alaikum. This is Sadia Habib from University of Agriculture, Faisalabad, Department of Zoology, Wildlife and Fisheries. So my topic for today's presentation is microbial analysis and hematological parameters of Labiorhita after dietary administration of black cardamom, Ammonium sublatum of different dietary percentages. So this is all about my abstract of the research. I'll discuss it later. If I talk about the objectives, so there were two major objectives of my research. The first one was to study the effect of Ammonium sublatum, known as black cardamom, kali lychee, on microbial analysis of Labiorhita of different dietary percentages, and secondly to check how Ammonium sublatum affect the hematology and RBC count of Labiorhita. If I talk about the importance of fisheries, we will know that अगर हम लोग 1950s की बात करें तो एक्वाकल्चर की जो प्रोडक्शन थी वो वर्ल्ड वाइड इंक्रीज हुई, लेकिन अगर हम लोग अर्ली 90s की बात करें या 1990s की बात करें तो कैप्चर फिशरीज में इतना इजाफा नहीं हुआ. ह्यूमन कंजप्शन की बात करें तो एक्वाकल्चर प्रोडक्शन जो है वो कैप्चर फिशरीज को आउटपेस कर गई, स्पेसिफिकली इन 2016 एंड 2018. बेसिकली 52% ऑफ टोटल वेट जो था एक्वेटिक क्रिएचर्स का जो ह्यूमन कंजप्शन के लिए यूज़ किया जाता था वो यहीं से ही आ रहा था. ये वाली अमाउंट जो है ये 60% तक इंक्रीज कर गई या इंक्रीज कर जाएगी इन 2015. इन द नटशेल अगर हम कहें तो ऐसी फिश प्रोडक्ट्स, ये कैप्चर फिशरीज जो हम लोग दरियाओं और समुद्रों से पकड़ते हैं वो इतनी ज़्यादा नहीं है. लेकिन अगर हम लोग एक्वाकल्चर की बात करें तो एक्वाकल्चर ने ह्यूमंस की वो डिमांड को मीट किया. बहरहाल अगर हम लोग एक्वाकल्चर की बात करें तो फिश मील और फिश ऑयल्स जो है बेसिकली इनकी प्रोडक्शन हैवीली इस चीज़ पर डिपेंड करती है. इनलैंड फिशरीज़ की बात की जाए जो कि मिलियंस ऑफ फिशरमैन को ना सिर्फ फूड सिक्योरिटी देती है बल्कि रोज़गार के मौके देती है, एम्प्लॉयमेंट जो है वो उनको प्रोवाइड करती है. इसके अलावा अगर हम लोग एक्वाकल्चर की बात करें स्पेसिफिकली तो देयर आर 425 स्पीशीज which are currently produced in aquaculture system and which is a very diverse number. उसके बाद जो हमारा मॉडल ऑर्गेनिज्म है जो हमने अपने रिसर्च में यूज़ किया विच इज़ रोहू लेबियोरोहिटा. लेबियोरोहिटा बेसिकली साउथ एशियन कंट्रीज़ में पाई जाती है और प्रोमिनेंट फ्रेश वाटर फिशेस को जो है वो बोला जाता है. इसके अलावा अगर जेनेटिकली देखा जाए तो इट इज़ मॉडिफाइड. बहुत हद तक रिसर्चर्स ने इस पे काम किया है. इसके गट माइक्रोबायोटा को देखा जाए और इसकी हर्बल रेमेडीज़ को देखा जाए तो इट इज़ द वे एडवांस्ड. उसके अलावा अगर इकोनॉमिक स्टैंड पॉइंट की बात की जाए तो बहुत सारे कंज्यूमर्स जो हैं वो इस स्पीशी में ना सिर्फ इंटरेस्टेड हैं बल्कि इन्वेस्टेड भी हैं. इसके ऊपर बहुत सारी रिसर्चेस हुई हैं और हाउएवर ये बहुत ज़्यादा डेवलप्ड जेनेटिकली मॉडिफाइड फिश है. इसके अलावा अगर हम फिश के गट माइक्रोबायोटा की बात करें विच इज़ वेरी सेंसिटिव पैरामीटर, तो ना सिर्फ साइंटिस्ट ने इसको कल्चर किया है बल्कि ये पता लगाया है कि बहुत सारे डिफरेंट बायोलॉजिकल प्रोसेसेस जैसे कि ग्रोथ, इम्यूनिटी, न्यूट्रिशन और जो है वो रिप्रोडक्शन इसके ऊपर अफेक्ट जो है वो डायरेक्टली इससे अफेक्ट होती है. उसके अलावा बहुत सारी फिशेस जो है वो अपने माइक्रोबायोटा को जो है वो चेंज करती हैं. अगर हम लोग हर्ब्स की बात की जाए, क्योंकि हर्ब्स को फ़िश की फीड में एज अ फीड एडिटिव यूज़ किया जाता है, मेडिसिनल पॉइंट ऑफ व्यू से भी ये ना सिर्फ फ़िश की इलनेस को रिमूव करती हैं बल्कि उसके एंटी ऑक्सीडेंट स्टेटस को इम्प्रूव करती हैं. इसके अलावा फ़िश में इम्यूनिटी भी जो है वो पैदा करती हैं. ब्लैक कार्डमम इज़ वन ऑफ द एग्जाम्पल ऑफ हर्ब्स. इट इज़ टॉल क्लस्टर्ड एवरग्रीन लाइक ब्लैक कार्डमम. इट इज़ ग्लोसी थिन ट्रॉपिकल फोलिएज होता है इसका. इसके अलावा इसका एंटीऑक्सीडेंट स्टेटस है और जो है ये फ़िश का एंटीऑक्सीडेंट जो है स्टेटस उसको ना सिर्फ इंक्रीज़ करती है बल्कि इसके अंदर बहुत सारे बेशुमार इम्पॉर्टेंट जैसे कि नियासिन, विटामिन सी, कॉपर, राइबोफ्लेविन वगैरह जो है वो पाए जाते हैं। अगर मैं मेथोडोलॉजी की बात करूँ तो हमारे फ़िश फार्म में यूनिवर्सिटी ऑफ एग्रीकल्चर फैसलाबाद में जो हमारा डिपार्टमेंट ऑफ ज़ूलॉजी, वाइल्ड लाइफ एंड फिशरीज थी, वहीं के ही फिश फार्म से वी कलेक्टेड अबाउट 80 फिश फिंगरलिंग्स वेटेड अबाउट लाइक 4.18 to 7.5 ग्राम्स। उसके अलावा हम लोगों ने एक्सपेरिमेंटल डिज़ाइन देखा तो देयर वर मेनली थ्री एक्वेरियाज, जिसमें हम लोगों ने फर्स्ट एक्वेरियम वाज प्रोवाइडेड विद T1 ग्रुप फेड विद जिसमें हम लोगों ने 2% ब्लैक कार्डमम एक्सट्रैक्ट डाला था, और जो T2 ग्रुप है इसके अंदर बेसिकली हम लोगों ने 30 फिशेस का ग्रुप लिया, इसको 1.5% ब्लैक कार्डमम के साथ ट्रीट किया। एंड द थर्ड ग्रुप विच वाज कंट्रोल ग्रुप, इट वाज विदाउट एनी लाइक कार्डमम एक्सट्रैक्ट और पाउडर। उसके बाद फीड प्रिपरेशन की बात की जाए तो इसकी स्पेसिफिक प्रोटोकॉल्स दे के अवन के अंडर हम लोगों ने इसको स्टेरलाइज करके फाइनली ग्रेन कर लिया और पाउडर्ड फॉर्म में जो है वो कन्वर्ट कर लिया। उसके बाद अगर मेथोडोलॉजी की बात करें, अगर हम लोग हीमाटोलॉजिकल पैरामीटर्स की बात करें विच इज़ वेरी वेरी मच इंपॉर्टेंट, जो कि एज हम जानते हैं कि ब्लड जो है वो कनेक्टिव टिश्यू भी होता है। कोई भी वाटर में चेंजेस आ रहे हैं मेजर या माइनर किस्म के, फ़िश के ब्लड से असेसमेंट की जा सकती है कि फ़िश की ओवरऑल बॉडी में किस तरह से चेंजेस आई हैं। सो उसके लिए हम लोगों ने MCH, MCHC, PCP, WBC एक्सेट्रा इन तमाम पैरामीटर्स को देख लिया। उसके बाद फ़िश के अगर गट में इंटेस्टाइनल गट माइक्रोबायोटा की बात की जाए तो देयर वर 3 डिफरेंट काइंड ऑफ जेल्स, न्यूट्रेंट एगर, ट्रिप्टिक सोय एगर, एओसिन मिथाइलिन ब्लू एगर। बेसिकली इसको यूज़ किया, इसका स्पेसिफिक इसको प्रोटोकॉल दिया, और प्रोटोकॉल के अंडर जो है वो हम लोगों ने स्पेसिफिक फार्मूलाज लगा के ये चीज़ काउंट कर ली कि फ़िश की जो ओवरऑल माइक्रोबियल काउंट है वो एक्सपेरिमेंटल ग्रुप्स और जो हमारे पास ट्रीटमेंट ग्रुप्स है इनमें इंक्रीज़ कर रहा है या डिक्रीज कर रहा है। वी विल फर्दर सी इट इन द रिजल्ट्स। उसके बाद फिजिको केमिकल पैरामीटर्स ऑफ वाटर्स की बात की जाए, जिसमें DO है, pH है, टेम्परेचर, वाटर, इन सबको हम लोगों ने रेगुलर बेसिस पे चेक किया था। स्टैटिस्टिकल एनालिसिस की बात करें तो हम लोगों ने उस डिज़ाइन को प्रॉपरली स्टैट लगाई, जिसमें द मोस्ट फिटेड एंड सूटेड वन वॉज द वन वे एनोवा स्टाइल। उसके बाद हम लोगों ने इसके ऊपर जब कंपैरिजन किया डिफरेंट ग्रुप्स का, तो हम लोगों ने पोस्ट हॉक टकी टेस्ट जो है वो उसको यूज़ किया ताकि दो स्पीशीज़ के दरमियान स्टैटिस्टिकल डिफरेंसेस का पता चल जाए। उसके बाद हीमाटोलॉजिकल पैरामीटर्स के अगर रिजल्ट्स की बात की जाए तो हम लोगों ने ये देखा, जो हमारे पास ट्रीटमेंट ग्रुप्स थे उनके अंदर WBC और RBC जो है दे वर नोटिसेबली हायर, एंड वी कैन सी इट इन द टेब्यूलर डेटा, जो कि उसको बहुत जो है वो इलेबोरेट वे में एक्सप्लेन कर रहा है। सो अगर ओवरऑल देखा जाए तो जो हमारे पास एक्सपेरिमेंटल ग्रुप था जिसमें हम लोगों ने नो कार्डमम ग्रुप था, उसके अंदर बेसिकली कमी आई, और जो हीमाटोलॉजिकल पैरामीटर्स थे T1 और T2 जिसमें हम लोगों ने कार्डमम, ब्लैक कार्डमम जो है 1.5% और 2% की रेशियो से दिया था, उसमें वाजिया इंक्रीज आया। सो दिस इज ऑल अबाउट द स्टैट्स एंड द रिजल्ट्स ऑफ स्पेसिफिक पैरामीटर्स। और ये हमारे पास सारी की सारी जो टेब्यूलर और ग्राफिकल रिप्रेजेंटेशंस है कि किस तरह से जो हमारे पास T0 और T1 और T2 जो ट्रीटमेंट्स थे फिशेस के, उनमें वाजिया इंक्रीज देखा जा सकता है एज कम्पेयर टू T0। उसके बाद अगर हम लोग इंटरनल माइक्रोबियल काउंट की बात करें तो जो गट सैंपल्स और माइक्रोबियल सैंपल्स हैं जिसमें हम लोगों ने डिफरेंट मीडियाज़ के अंदर उसको ग्रो करवाया था, जिसमें सबसे ज़्यादा रिचर जो डाइवर्सिटी है वो हमारे पास जो है वो TSA में, ट्रिप्टिक सोय एगर में देखी गई, और जो हमारे पास EMP है वो सिर्फ नेगेटिव ग्राम नेगेटिव बैक्टीरियाज को ही सपोर्ट करता है। खैर इन द नटशेल ये देखा गया कि जो हमारे पास ग्रुप्स थे जिनको ट्रीटमेंट ग्रुप्स कहा जाता था, उनके अंदर कमी आई थी माइक्रोबियल काउंट को लेके, जबकि एज कम्पेयर टू एक्सपेरिमेंटल ग्रुप, बिकॉज हमारी जो फीड है, जो ब्लैक कार्डमम है, इट एक्ट एज एन एंटीऑक्सीडेंट और एंटी माइक्रोबियल। तो ये क्या करती है, फिश के इम्यून सिस्टम को ना सिर्फ बूस्ट करती है बल्कि जो हमारे पास बैक्टीरियाज हैं, स्पेसिफिकली हार्मफुल बैक्टीरियाज हैं, जिसमें हमारे पास एरोमोनाज और दूसरी स्पीशीज आ जाती हैं, तो उनमें जो है वो एक डिक्रीज देखा गया था। और जो हमारे पास एडवांटेजेस जो बैक्टीरियाज आते हैं जिसमें लैक्टोबैसिलस और बैसिलस वगैरह होते हैं, इनमें इंक्रीज देखा गया था। तो एरोमोनाज और सोडोमोनाज जैसे हार्मफुल बैक्टीरियाज को इस फीड ने However, the results are far clear. जैसा कि हम लोग ये टेबुलर फॉर्म और स्टैटिस्टिकल फॉर्म से भी इस चीज़ को बहुत क्लियरली देख सकते हैं. द वे वी अप्लाइड द स्टैटिस्टिक्स एंड उसके बाद हमारे पास स्टैंडर्ड डेविएशन और जो दूसरी वैल्यूज़ हैं वो वाज़े तौर पे इस चीज़ को बता रही हैं. जैसे ये हम लोग ग्राफिकल रिप्रेजेंटेशन भी देख सकते हैं चीज़ों की, तो इससे अंदाज़ा ये होता है कि जो हमारे पास T0 वैल्यू था, लाइक जो एक्सपेरिमेंटल ग्रुप था, इसमें इंक्रीज था. इट वॉज अ कंपैरेटिव इंक्रीज, या दूसरे मायनों में ये भी कहा जा सकता है कि जो हमारे पास ट्रीटमेंट ग्रुप था उनमें जो उनका माइक्रोबियल काउंट था, इट वॉज द वे डिक्रीज्ड कंपैरेटिवली. खैर उसके बाद इन द नटशेल अगर हम लोग कंक्लूजन की बात करें तो जो हमारी स्टडी की फाइंडिंग्स हैं वो इस चीज़ को वाज़ह तौर पे बताती हैं कि जो ब्लैक कार्डमम है वो बहुत सारी इंपॉर्टेंट डिटेल्स को बताता है. इसमें इट इज़ अ वेल्थ ऑफ मिनरल्स, फिनोलिक, एंटी माइक्रोबियल एंड एंटीऑक्सीडेंट प्रॉपर्टीज़. इसके अलावा जो डिफरेंट स्टडी टाइप्स थी बैक्टीरियाज की, वो इस चीज़ को बताती हैं वाज़ह तौर पे कि इट इज़ अ एंटी माइक्रोबियल फीड. उसके अलावा अगर हम WBCs और RBCs काउंट की बात करें तो इस फीड ने जो है वो उनके हीमाटोलॉजिकल पैरामीटर्स को इंक्रीज़ किया, उसकी इम्यूनिटी को बूस्ट किया. इसके अलावा जो है वो फ़िश की डाइट को होल जो है वो एक एज मिला इससे. हाउएवर इन द नटशेल वी वुड कंक्लूड दैट ब्लैक कार्डमम्स inclusion in raw diets offer promising opportunities for sustainable aquaculture. However, it was all about our research. इन द नटशेल ये कि वो जो हमारी रिसर्च थी जिसमें हम लोगों ने फ़िश को 2 मंथ्स का ट्रायल दिया और उसके बाद उसको डिफरेंट ट्रीटमेंट्स के साथ हम लोगों ने जब डिफरेंट ट्रीटमेंट्स दिए डिफरेंट डाइटरी परसेंटेजेस के साथ, तो ये चीज़ बहुत वाज़ह तौर पे सामने आई कि इट वाज अ पॉजिटिव चेंज बाय द डाइट in their overall functioning. Thank you very much.
Thank you, Ms. Sadia, for that informative contribution to fisheries biochemistry. Our fourth speaker is Ms. Laiba Akhtar from the University of Veterinary and Animal Sciences. She will present on effects of biotin supplementation on growth, proximate composition, glycogen content, and enzyme activities of Labiorhita fed with high carbohydrate diet. Welcome, Ms. Laiba Akhtar.
Assalamualaikum, myself Laiba Akhtar, and the topic is effects of dietary biotin on growth, proximate composition, glycogen content, and enzyme activities of Labiorhita fed with high carbohydrates diet. Table of content. Introduction. Aquaculture is a rapid growing form of food production across the worldwide. Aquaculture production depends on fish feeding under favorable conditions. Fish feed is typically made up of proteins 18 to 50%, lipids 10 to 25%, carbohydrates 15 to 20%, ash more than 8.5%, phosphorus more than 1.5%, water more than 10%, and trace amounts of minerals and vitamins. Proteins are the most expensive component of fish feed, whereas carbohydrates are the affordable energy source. Carbohydrates are cheap and easily available in markets, still not preferred as major feed ingredient as protein because of their low digestibility in fish as compared to other animals. It starts to accumulate in the body of fish that has negative effect on fish. Furthermore, high levels of carbohydrate causes severe reactions in fish such as weak immunity, high mortality, less growth rate, and less nutrient consumption ratio. Biotin is a component of vitamin B complex that is essential for animal growth and acts as enzymes cofactor involved in the metabolism of carbohydrates, proteins, and lipids. Biotin has a lot of positive effect on fish such as growth rate, weight gain, survival rate, protein conversion ratio, and glucose metabolism. Deficiency of biotin leads to reduced appetite, slow growth, atrophy, and feed consumption in various species like Japanese sea bass, grass carp, and giant carp. Literature reports that biotin in high carbohydrate diets increase the digestibility of carbohydrate and reduce their negative effect in animals. Statement of problem. Carbohydrates are cheap and easily available in market, provided enough energy for growth and development. Still, it is not preferred as fish feed because of their low digestibility. So it starts to accumulate in the body of fish that has negative effect on fish. Biotin is a vitamin that is essential for growth. Fish with biotin show high growth rate, high appetite, and low mortality, and have ability to produce insulin receptor to reduce hyperglycemia in fish. Biotin supplemented in fish feed can reduce the harmful effect of high carbohydrate supplementation in fish. Objectives: to analyze the effect of dietary biotin on growth, biological indices beside body composition of high carbohydrates fed laboratory. To analyze the activities of digestive enzyme, antioxidant enzymes, peptic enzyme, and MDA level. To investigate the effects of carbohydrates and biotin on plasma glucose, glycogen in liver, glycogen in muscle, and biotin level in plasma and liver of fish. Hypothesis: supplementation of biotin may reduce the harmful effects caused by high carbohydrate diets and improve the growth and health of laboratory. Materials and methods. Materials and methods experimental area. This study was conducted at Fish Seed Rearing Unit of Department of Fisheries and Aquaculture, University of Veterinary and Animal Sciences, Ravi Campus, Pattoki. Composition of basal diet. Ingredients: soybean meal, fish meal, sunflower meal, cornstarch, fish oil, biotin free, vitamin premix, mineral mixture, biotin, crude protein, and nitrogen free extract. Experimental design. Basal diet containing 35.12% of the carbohydrates. Biotin 6 experimental diets prepared, named as B0, B0.15, B0.3, B0.6, B1.2, and B2.4. Formulation of diet. Dry ingredients ground and screened in grinder. Add mineral premix, fish oil, and vitamin premix. Add water, make dough, place in meat mincer, pellet form, and dry them and store in vacuum bags. Experimental fish would be Laborahita. Fish husbandry. Fingerlings of Laborahita acclimatized for 2 weeks, stock 15 fish in each replicate, fed with test diets twice a day, pH, adjust pH, temperature, and dissolved oxygen, continue for 60 days. Fish harvesting. At the end of the experiment, starve the fish for 24 hours, anesthetize the fish, and whole body plasma, intestine, liver, and muscle samples collected. Growth performance. Weight gain in percentage, FCR, survival rate in percentage, and SGR would be determined by using these formulas. Proximate analysis. Crude protein was determined after the determination of nitrogen on gel apparatus after acid digestion. Crude fat was determined by ether extraction through Socklet apparatus. Crude ash was determined by flaming the sample at 600 degrees Celsius. Moisture content was determined by drying sample at 105 degrees Celsius to a constant weight. Body indices. Hepatosmetic index and visosmetic index would be can be determined through by using these formulas. Plasma biochemistry. Blood was taken, separate plasma at 300 rpm. Arena bioscience kit was used. Biotin in liver and plasma. Fish were dissected, separate liver and plasma sample. Biotin assay kit was used to analyze the samples. Glycogen content in liver and muscle. Fish were dissected, separate liver and muscle. Glycogen assay kit was used to analyze the sample. Digestive enzyme activities. Fish were dissected, separate intestine sample. Amylase activity by Burnfield method. Lipase activity by Chare and Crandall method. And protease activity by Gracio's Carino method. Antioxidant enzyme and MDA level. Catalase activity was determined by following Chance and Mehaly method. Superoxide dismutase activity was determined by following Ganopolitis and Rice method. Glutathione peroxidase activities was determined by following Civello method, whereas MDA level was determined by following Gata method. Statistical analysis. One ANOVA was used for data analysis. Broken line regression analysis was performed to assess the optimal dietary biotin requirement of Laborahita. If significant occurs, honestly significance difference test was applied for comparison of means. Results: growth performance, growth parameters, and feed utilization. Increase in dietary biotin significantly improved growth performance, weight gain, and feed efficiency with highs value at B0.60 and FCR decreased at B0.60. That shows that increase in biotin can enhance the growth of the fish. Graphical representation of optimal value of Laborahita. The optimal value of Laborahita is determined by this method is 0.55 milligram per kg. Body indices. Visosmetic index and hepatosmetic index remain stable with no significant differences when fed with different biotin levels. Proximate analysis. Innate moisture CP, CF, AC has it remains stable with no significant differences when fed with different biotin levels. Digestive enzyme activities, amylase, lipase, and protease activity increases as the biotin level increases, and maximum growth were observed at B0.60. Antioxidant enzyme and MDA level. Enzyme activities were maximum at B0.60 as shown in table, and MDA level is low at B0.60. Glycogen and biotin determination. In liver glycogen, biotin had no effect on liver glycogen level, whereas in muscle glycogen, biotin did not influence muscle glycogen storage. Whereas in plasma biotin, dietary biotin significantly affect plasma biotin, and liver biotin content increase markedly, showing effective absorption and storage in fish. hepatic enzyme activities, and plasma glucose. ALT and AST decreases at B0.60, whereas ALP activity increases at B0.60 as given in table. Similarly, plasma glucose level decreases at B0.60. Conclusion. This study examined the impact of dietary biotin supplementation in high carbohydrate diet on labiraheta. The optimal diet, biotin level of 0.60 mg per kg significantly enhanced growth, performance, feed efficiency, antioxidant defense, and liver function. At the supplementation level, improvements were seen in weight gain, feed conversion ratio, and enzyme activities, including digestive and antioxidant enzymes. Plasma and liver biotin levels increased with supplementation, leading to better management of oxidative stress and reduced MDA level, a marker for oxidative damage. Biotin supplementation at 0.60 mg per kg showed promise in enhancing growth, feed utilization, enzymatic functions, and oxidative stress management in labiraheta. These results underscore the effectiveness of biotin as a dietary supplementation of boosting aquaculture productivity. Suggestions. This study will provide a cost-effective feed to farmers because high carbohydrate will spare the protein, which is an expensive constituent of fish feed. Thus, it will ultimately reduce the production cost in aquaculture industry. Literature citation. Thank you.
Thank you, Ms. Laiba, for those interesting findings on nutrient supplementation in fish. The next presentation is by Ms. Aisha Imran from the University of Sargodha. Her research focuses on biomonitoring of selected heavy metals accumulation in Indian Manna eccritotheristis from district Narowal. Please welcome Ms. Aisha Imran.
Bismillahirrahmanirrahim. My name is Aisha Imran from University of Sialkot and from Department of Zoology. The title of my research is Biomonitoring of Heavy Metal Accumulation in Indian Minor eccritotheristis from district Narwal. Here is the topic: table of content, graphical abstract, introduction, problem statement, objective, material and method, metal analysis, statistical analysis, results, and reference. The graphical abstract of my paper, which explains the summary of my paper. Introduction. Toxic metals are rising concern for human, animal, and environmental health. Industrialization, mining, agriculture, and urbanization are main sources of pollution. Heavy metals are resistant to things, resistant to degradation, and accumulate in ecosystem. They move through the food chain, concentrating in high trophic level, including blood. Toxic effect of heavy metal. In small amounts, some metals are tolerated or even essential for our body, such as zinc, copper, etc. Excess exposure causes pathological effects due to protein, DNA, and cell membrane. Heavy metal disrupt enzymatic processes, leading to genetic mutation and cell dysfunction. Birds exposed to metals suffer reproductive failure, neurological damage, and immune suppression. Indian Myna. The Indian Myna eccritotheristis belongs to the Stridoniid family and is widely distributed. It was introduced for pest control. It is now invasive and abundant across continents. It is also classified in IUCN Reduced and IUCN List as least concern, but considered one of the most invasive alien species. Due to abundance, adaptability, and close association with human habitat, it is a strong bioindicator. Heavy metal and burns. Common toxic metals. Here is the list of common toxic metals such as lead, cadmium, arsenic, mercury, nickel, chromium. Essential metals in excess also become toxic. Birds absorb metals through contaminated food, water, air, or direct contact. Metal accumulate in feeder, liver, kidney, lungs, and eggs. High concentration interfere with physiology, metabolism, growth, and reproduction. Feeders as bioindicator. Feeders are excellent and invasive bioindicator for metal contamination. They reflect metal accumulation during feeder growing, providing long-term exposure history. Higher concentration often found in feeder compared to other tissues. Feeder analysis is widely used in ecotoxicology to monitor environmental methylation trend. Example, let X-square inhibit pH enzyme disrupting hemoglobin synthesis in plants. Here is the sources of X-square: feeding, drinking, and respiration. Problem statement: the Industrial Revolution has led to the accumulation of heavy metal in the ecosystem, posing a significant threat to bird species and affecting their behavior, health, and population dynamics. These are the objectives: to assess heavy metal concentration in feeders, kidney, and liver of medium level. The second objective is to evaluate the difference of heavy metal concentration in different habitat types of normal tissue. Here is the material of method. There is the following material that we had been used for performing our research. Study area. The map of my study area district R1, which has 3 essays: sugar and sulfur and lime. Experimental view, sample collection, addition of sample, and method analysis. Experimental design, sample collection of liver, kidney, and liver. There is the flow sheet of from where I was given samples. Digestion of organ, liver, and kidney for heavy metal. The first step is dry the samples. After that, weigh the samples, and then digestive reagents were added into the samples, which has 2 nitric acid and hydrogen peroxide. After that, the digestion process were done on hot plate until the mixture turned pale yellow. Then primary dilution were done with diluted water, 25 ml deionized water. Then filtered the sample to remove impurities. After that, the final dilution were done with 50% distilled water. Then the final step is store the sample at 4 degrees in sample bottles for further metal analysis. The next is digestion of feeder samples of heavy metals. The first step is washing feeder samples with distilled water and 1 molar acetone 3 times. After, the second step is air drying. Air dried the samples to remove surface moisture, then oven dried for 2 to 3 hours at 105 degrees, and then cutting the samples. Then we will cut the samples into small pieces by stainless scissors. With stainless scissors, by choosing stainless scissors. The next step is digestive reagent. The 0.5 grams are feeder. After the weighted, added the reagent. Then the more reagent is 0.25 ml of chloric acid and 1.0 ml of nitric acid were added into the feeder samples. After that, cooling the samples. After heating the samples at room temperature, and then filter the samples with 50 ml of distilled water. After the filtration, the samples stored at room temperature containers for further metal analysis by spectroscopy. Here is the metal analysis and used coupled plasma spectrometer for determination of metals. The statistical analysis, and then results. Here is the results. The p-value in feeders, zinc 0.01. It means it's significant, highly significant. Then zinc in kidney 0.03, and zinc in liver is mostly significant because the p-value of zinc in liver is 0.000. Like zinc, here is the chromium, and chromium shows the negative value in site 1. Then chromium in kidney, liver, feeder, cobalt in kidney, liver, feeder, and then all the other heavy metals. Here is the summary of all the heavy metals and with their P values. The reference. That's all about my presentation. Thank you.
Thank you, Ms. Aisha, for this wonderful talk. Our next speaker is Ms. Aiza Munawwar Ali from University of Agriculture, Faisalabad, presenting on effect of fructooligosaccharides and xylooligosaccharide on growth and hematology of hypophthalmictus mollitrix. Please welcome Ms. Aiza Munawwar Ali.
Assalamualaikum. I have selected the topic for my thesis is effect of fructooligosaccharides and xylooligosaccharide on growth and hematology of hypophthalmictus mollitrix. The objectives of my thesis is to investigate how fructooligosaccharides and xylooligosaccharide affect hypophthalmictus mollitrix hematological parameters and to determine how fructooligosaccharides and xylooligosaccharide enhances the growth performance in hypophthalmictus mollitrix. Aquaculture is the procedure of raising aquatic floras and faunas including fish, algae and shellfish in regulated settings is called aquaculture, and aquaculture has grown speedily in recent years with total production rising by 32% between 2010 and 2018 according to FAO. Aquaculture is one of the most active areas of the global food system and a valuable supply of important dietary elements for the world's population. Fisheries directly support the nation's food supply, provide a means of subsistence for those living along the coast, generate revenue from exports, and strengthen the economy. Fish is an excellent source of vitamin A, D, calcium, omega-3 proteins, iron, and other nutrients that can help treat a variety of diseases. A range of inland and marine fisheries is represented by Pakistan's natural resources. Inland fish are found in Punjab, Sindh and KPK, while marine fish are found in southern region of Pakistan including Sindh and Balochistan. Silver carp is a freshwater species that is naturally found in reservoirs, lakes, streams and ponds. China are utilized this fish to control cyanobacteria blooms because of their filter feeding behavior. It contains high protein content and makes a substantial contribution to the output of freshwater aquaculture worldwide. The overall morphology of hypophthalmictus mollitrix described as having a deep body and being somewhat compressed. They are silver in color with a darker silver back and a white belly. But with age they gain an olive green cast over their backs. Fructooligosaccharides and xylooligosaccharide both are prebiotics and act as non-digestive, anti-freezing and non-carcinogenic activities. The trial materials and methods. The trial was held at Fisheries Research Farm at the Department of Zoology, Wildlife and Fisheries. The trial duration was 2 months. The trial was started from September 18, 2024 and ended on November 6, 2024. I have taken 60 healthy hypophthalmictus mollitrix samples with an average weight of 3 to 5 grams. Fish stocking. Fish were taken from a study pond and put in 2-glass aquarium for the experiment. In the lab, 2-glass aquaria with 20 hypophthalmictus mollitrix fingerlings. Each were set up as the experimental group and the control group. The experimental group T1 were fed with fructooligosaccharides and T2 were fed with xylooligosaccharide, and control group were fed with commercial feed. I have taken fructooligosaccharides and xylooligosaccharide from market. Assessment of growth performance. Throughout the trial period, a variety of growth measures were assessed to gauge the fish progress. These measurements include body weight in grams and length in centimeter. Crucial metrics like as survival percentage, feed conversion ratio, and specific growth rate were computed as well. Determination of hematological parameter. A hematocytometer were used to measure the total red blood cells and white blood cells counts for blood chemistry. Initially, the blood sample was separated into 2 sections. Hematological indexes such as hemoglobin, hematocrit, red blood cells and white blood cells were determined in the first section using 10% EDTA. A 100 microliter blood sample was incubated in micro flat bottomed plates for 60 minutes at 30 degrees Celsius, primarily to facilitate cell adhesion. The saline buffer will then be utilized for washing. We were washed 3 times in order to obtain a precise count. This approach was used to calculate the hematological parameters including mean cell volume and mean cell hemoglobin. Physicochemical parameters of water. On each trial day, a number of significant limnological parameters including pH, dissolved oxygen, temperature, total hardness, total alkalinity, and salinity of water were assessed. Temperature, dissolved oxygen, and pH levels were measured using electronic instruments such as HANA HI 8424 and a pH meter. The water hardness, salinity, and total alkalinity were measured using standard methods. Statistical analysis. The CRD ANOVA was used to compare the outcomes and get at the ultimate interpretation at the conclusion of the experiment. Results. Limnological parameters. Now you can see the progress of T2, which were fed with xylooligosaccharide as compared to T1 and T2. Impact of fructooligosaccharides and xylooligosaccharide on growth performance of hypophthalmic thismolytics. T2, the growth performance of tank 2 were much better than T1 and T2. The results of T2 were much better than T1 and T2. Average weight of T2 were higher than T1 and T2. Weight gain, you can see weight gain of T2 as compared to T1 and T2. Overall, the results of growth performance of T2 were much better than T1 and T2. Now you can see effect of fructooligosaccharides and xylooligosaccharide on the hematological parameter of hypophthalmic thismolytics. T2, red blood cells of T2 which were fed with xylooligosaccharide as compared to T1 which were fed with fructooligosaccharides and T2 which were fed with commercial feed. Red blood cells, white blood cells, hemoglobin and hematocrit are parameters of hematology. The overall results of growth performance and hematology were highly significant. Now you can see silver carp. Discussion. The experiment was conducted at the Microbiology and Immunology Laboratory, Department of Zoology, Wildlife and Fisheries, University of Agriculture, Faisalabad. The research was carried out to assess the effect of fructooligosaccharides and xylooligosaccharide on growth and hematology of hypophthalmic thismolytics. Fructooligosaccharides act as prebiotic, giving the beneficial bacteria in the stomach an indigestible food source. It has been demonstrated that fructooligosaccharides promotes a healthy gut microbiota and raises levels of lactobacillus and bifidobacterium. Xylooligosaccharide can help improve blood chemistry, growth performance, and the manufacture of lipid metabolites, which can give fish intestinal lining cells energy. My results. Were alike more shady. So more shady examined the positive effects of dietary xylooligosaccharide on the body structure, blood, immune processes, and development presentation of juvenile Sobitis C. bream. Zhang et al. 2020 studied the effects of xylooligosaccharide in juvenile grass carp diets, improved ultimate body weight, weight increases, and specific growth rate parameters without negativity impacting health or feed efficiency. Likewise, in the current research on hypophthalmic thermometrics, higher mean values of growth performance than the control group when was completely substituted with xylooligosaccharide feed. Conclusion: the findings of this investigation show that xylooligosaccharide improves hypophthalmic thermometrics, hematological, and growth performance. The hematological of hypophthalmic thermometrics improve more under xylooligosaccharide, leading to improved FCR and higher SGR, weight gain, length gain, and survival rate. Thank you.
Thank you, Ms. Aiza, for that insightful presentation on dietary prebiotics in aquaculture species. Next, we have Ms. Samra Riaz from University of Agriculture, Faisalabad. Her topic is toxicological effects of chromium chloride on histopathology of labiorrheta. Please welcome Ms. Samra Riaz.
Assalamu alaikum everyone. I am Samra Riaz, a postgraduate student from the Department of Zoology, Wildlife and Fisheries at the University of Agriculture, Faisalabad, Pakistan. Today I am presenting on the toxicological effect of chromium chloride on histopathology of labiorrheta at the 8th International Conference on Applied Zoology 2025. In the introduction, I would like to highlight a few key points that set the stage for my research. Aquaculture is one of the most important areas of food production globally, playing a vital role in meeting the increasing demand for protein-rich food. Asia leads the world in aquaculture production of fish, accounting for its 6.3% of global aqua production. This is significant contribution underscores the importance of maintaining healthy aquatic ecosystems for sustainable food production. However, over the last 20 years, aquatic pollution has become a significant environmental issue. The impact of pollution on aquatic life and the overall ecosystem cannot be overstated. Waste from industry is the main cause of heavy metal pollution in aquatic environments. Heavy metals are a big concern in this context because of their toxicity and persistence in the environment. Heavy metals by definition are any metallic elements with a high density that are hazardous even in trace levels. Common heavy metals include chromium, lead, arsenic, copper, cadmium, zinc, and mercury. These metals tend to collect more quickly in organisms and are challenging for living organisms to digest or dispose of. This bioaccumulation can lead to severe toxicological effects on aquatic life, including fish like labiorrheta, which is the focus of my study. Urbanization and extensive industrialization are the main causes of pollution in aquatic ecosystems. As industries grow and urban areas expand, this risk of heavy metals pollution in water bodies increases, posing a threat to aquatic life and potentially to human health through the food chain. In my research on the toxicological effect of chromium chloride on histopathology of labiorrheta, I aim to understand how exposure to these heavy metals affect fish tissues and overall health. This understanding is crucial for assessing the risk associated with heavy metal pollution in aquaculture and for developing strategies to mitigate these impacts. Moving on the specifics of chromium, let's see some key points about this metal. Chromium is the sixth most common metal in the earth crust. It is widely used in various industries including the processing of medicines, textiles, printing press, process of dyeing, stainless steel, rubber and photographic printing industries. Chromium, particularly its hexavalent and trivalent forms, is found in the environment. The presence of chromium in aquatic environments is a concern because it causes structural alterations that can disturb several physiological processes. Ultimately, this can inhibit growth and lead to histopathological alterations in aquatic organisms like fish. Understanding the impacts of chromium on aquatic life is crucial for assessing the risk associated with heavy metal pollution in aquaculture and for developing strategies to mitigate these effects. The main objectives of the current study were investigation of toxic impacts of chromium chloride on Labiorhita, evaluation of different histopathological changes in Labiorhita exposed to chromium chloride in various organs like gills, liver, heart, and muscles. The study followed these steps for histopathological analysis. First one is tissue preparation. Preparation of tissues from Labiorhita for histopathological alterations or examinations. Fixation. Fixing the tissues. Preserve their structures. Dehydration and clearing. Dehydrating tissues and clearing them for further processing. Then impregnation. Impregnating tissues for embedding. Fifth one is embedding tissues in a medium for sectioning. Then the step comes section cutting sections of the embedded tissues. The last one is mounting and mounting sections on the slides and staining them for microscopic examinations. Results. I briefly discuss how fish serve as bioindicator of water pollution. Their capacity to absorb organic waste and pollutants makes them crucial for assessing water health. First of all, we will see effects on gill. Research shows that exposure to pollutants leads to widespread necrosis and degeneration of gill tissues. Thus, this disruption of gill structures can severely impact fish respiration and overall health. In terms of cardiac health, pollutants cause significant damage. Cardiac myofibrils show disarray and edema, along with focal vacuolar degeneration. This results in severe myocardial degeneration and necrosis. In conclusion, we can say fish are effective bioindicators due to their absorption of pollutants. The damage to gill and cardiac tissue highlights the need for monitoring water quality to protect aquatic life. Severe hypertrophy, edema, and necrosis in the liver. Severe hypertrophy, edema, and necrosis were seen. Splitting and degeneration of muscle fibers along with mild swelling of myocytes were observed. Sections revealed muscles elongation, severe muscle fiber degeneration, and necrosis. Extensive inflammation, congestion, and hemorrhage along with widespread necrosis and apoptosis of hepatocytes were seen in the liver. Loss of sinusoidal spaces and formation of fibrotic tissues were also observed. Conclusion. Animal histopathological alterations, particularly in fish, are strong markers to prior exposure to aquatic environmental stresses. Fish may experience stress from toxic chromium, which can impair a number of biological processes and result in certain histopathological changes. Fish tissues may exhibit macroscopic abnormalities and histopathological alterations due to metal deposition. Thank you so much for your time.
Thank you, Ms. Samra, for presenting valuable data on the toxicological effects of chromium. Our eighth presenter is Ms. Rabia Nazir from University of Agriculture, Faisalabad. Her presentation is titled Potential Role of Dried Fenugreek Seed Supplemented Diet on Growth Performance, Feed Utilization, and Survival of Hypophthalmictus mollitrix and Oreochromis niloticus. Let's welcome Ms. Rabia Nazir.
Assalamu alaikum. My name is Rabia Nazir. I am an MPhil scholar from University of Agriculture, Faisalabad. Today, I am excited to present my research work on potential role of dried fenugreek seed supplemented diet on growth performance, feed utilization, and survival of hypophthalmic dysmoletrix and Oreochromis niloticus. Aquaculture is recognized as one of the fastest growing sector in global food production, contributing significantly to food security, poverty alleviation, and economic growth. With the increasing demand for animal protein, aquaculture has become a vital source of high quality fish protein. However, one of the major challenges faced by the industry is to achieve sustainable growth while the maintaining fish health, survival, and growth performance. Feed costs account for a substantial portion of aquaculture expenses, and the efficiency of feed utilization directly influences the profitability. Therefore, the search for effective, affordable dietary supplement that enhance growth and reduce feed conversion ratio remains a priority. Now we see that the number of wild fish stock is declining at an accelerating rate. The simplest way to quarantine that there is enough seafood in the world is through aquaculture. Aquaculture has encountered significant challenges like limiting number of improved species, labor intensiveness, and environmental contamination, illnesses and disease resistance, etc. Currently, aquaculture farming relies the medicinal or over prophylactic use of antibiotics and feed additives to reduce disease load and productivity losses in fish kept in highly stocked and stressful environment. The rise of infection in aquaculture is related to careless use of antibiotic, which could prevent an issue worldwide for prevention and control of diseases either now or in future. Thus, looking for the safe, herbal, toxic-free antibiotic substitute is the interesting field of research having great potential to improve fish farming sustainability and security. One popular tactic is to utilize the immunostimulant to enhance the farmed aquatic organisms' immune-mediated condition. Herbal extract, phytochemicals, and plant candy metabolites are essential feed additives in fish farming that either boost the immune system, then finally their health, and then finally their survivability. The impact of plant supplements upon cells, human responses, and adaptive immune responses of fishes have been a recent push in study to look at how phytoadditives impact intracellular as well as the anatomical DNA expressions processes and then finally their growth. Numerous studies have shown that using plant with medicinal properties and their extract enhance fish performance, health, and functional role of various plant materials such as fenugreek, coffee beans, ginger on the defense system, and disease resistance and growth efficiency has been the objective of numerous research. But today in this research we discuss about the effect of plant-based feed additives or fenugreek on the defense system, growth, feed utilization, and survival of Nile tilapia and silver carp. Fish have been administered plant-based remedies to reduce the harmful influences of environmental factors and tension of toxicant. Now we discuss about the experimental design. In this experiment, we use two treatment groups, T0 group and T1 group. T0 group in T0 group we use commercial feed and we given the name control group, and T1 treatment group we fed only the dried fenugreek seed supplemented diet. In experiment design, Silver carp on tilapia fingerlings were gathered from the University of Agriculture Fishery Research Farm. And for a week before the experiment start, fingerling was kept in a tank to acclimate to the lab conditions for this setting. For 2 months or 60 days, the experiment was conducted in two glass aquarium with 20 fingerlings of Nile tilapia and silver carp of the same size housed in each aquarium, and regular water exchange help to maintain the quality of the water. Measurements such as body weight, length gain, weight gain, specific growth rate, that is FGR and FCR, feed conversion ratio, and survival percentages were among the growth parameters that can be checked at the end of the experimental period. Now we discuss about the feed preparation. That is fenugreek seed brought and processed at the neighborhood market. The first step, making a paste of each type, mixing it individually with 100 milliliter of warm water. A customized paste extruder was used to pelletize the paste after they had been processed in a grinder to make the diet understudy. Now we see the statistical analysis that shows I choose the independent t-test. Importance of independent t-test. Independent t-test was used to compare the means of two groups, or ANOVA was used more than two groups. That's why we choose the independent t-test. And now let's see the result of these two groups. As the result clearly show the difference between the T0 and T1 group, there is a significant difference or higher significant difference. Significant difference means the two groups are different from each other, both T1 and T0. There is a significant difference between two groups of two species, but now we see the difference of two species. When we see the difference between the two fishes, then the Nile tilapia has the higher growth than the silver carp. That may be due to the higher adaptable or better digestive system of Nile tilapia, or maybe it's more resilient than the Nile tilapia. When we see the cumulative effect of both species, we clearly see that both groups have higher growth than the T0 group, that is control group, that is given only the commercial feed. In conclusion, we say that this study offers important new information about the applications of fenugreek seed supplement in aquaculture, or contribute profitability, sustainability of fish farming method by boosting growth rate and nutrient utilization efficiency. At last, my many thanks to my supervisor Dr. Shakila Parveen for helping me out in my research, and at last many thanks to ICAZ 2025 for providing me this opportunity to present my research work. Thanks.
Thank you, Ms. Rabia, for sharing your research findings on herbal supplementation in aquaculture. Finally, we have Ms. Unza Seher from University of Agriculture, Faisalabad, presenting on effects of oral administration of Morus alba leaf powder on gut microbiota and survival indices of Hypophthalmictus mollitrix and Syrinus margalla. Please welcome Ms. Unza Seher. Thank you, Ms. Unza, for concluding our session with such a valuable presentation on nutritional microbiology in fish. With this, we conclude our session. I would like to extend my sincere thanks to all the presenters for their excellent contributions and to our audience for their engagement and attention. I also acknowledge the efforts of the organizing committee of ICAZ 2025 for providing this wonderful platform for scientific exchange. Thank you all once again.